welcome back, Life Sciences. I hope you just took a little bit of a stretch, a little bit of an exercise, got that blood pumping and the brain going, and we're ready to do more work on bacteria. Okay, so now we've looked at the structure, we've looked at the whole concept of what bacteria are. We just briefly went over a few diseases that you need to have, a good knowledge of. And now we're going to look at kind of questions you might find when you write your test or your exam. Okay, let's start with the questions. Okay, the first one, I always like to start off with a multiple choice question because the answers are there and it's nice to just get everything started. Okay, so what does the question ask us? And it's got quite a few big words in it, so we've got to try and make sure that we understand, all right, what we're doing. The question is, what are the causative agents? And another word we could use there, what causes? And I could put there, and which microorganism? Okay, and method of transmission. It means how does it spread? That's what we're looking at, okay, of tuberculosis. So they're asking you what microorganism, right, causes tuberculosis, and the second part is how does it spread? Right, so we've got various options here, and they need to manage, this must match, okay, column A and column B, so to speak, must match. Okay, so number A is bacterium. So is tuberculosis caused by bacterium? Yes, it is. And airborne droplets by sneezing or coughing. Right, and the answer is yes. But we're just going to go and check. So I'm going to circle A, because I think that is going to be the right answer. But I'm just going to check my other answers just in case, because B also is a bacterium, but it says it gets spread in water. And the one that we looked at was cholera, not TB. So that was incorrect. So it's not going to be B. Okay, C and D is a virus. And by now, right after today's lesson, tuberculosis is not caused by a virus, so that automatically says definitely not, C and D. So our answer definitely is going to be A. Okay guys, the next diagram, or the next couple of questions, are going to be related to a diagram of the bacteria. As I said to you, one of the things you must be able to do is label the bacterium cell and know the functions of the different parts. So what does this, all right, what is this question asking us for? So here is the diagram, and it says the diagram shows a cholera bacterium. So it's telling us that it is cholera. It also is giving us a little bit of information, all right, over there, that you are going to need to maybe do some kind of calculation, that it's magnified 50,000 times. Okay, so that whole little bit over here, there's lots of information in the title, and here is the diagram that we're going to have to use. How do I know it's a bacterium? Right, it's a single cell. Yes, I can see it has a tail, and I can see a round like nucleus. Right, so what are the questions? It says A, F, and G. So I'm going to circle them. A, F, and G. Right, are all parts of the boundary layer. What is the boundary layer? So another word is the outer layers. And remember I said to you, there are 3 outer layers that a bacterium cell will have. Now they've asked you to label them. Okay, so you can't just randomly label them. You need to know from the inside to the outside. Now A is pointing to the very inner one. So my A would be the cell membrane. That's the very first layer from the inside. G is pointing to the second one. That would then be my cell wall. And my final is F, and that is the round, let's look a little bit squidgy, and that would be my capsule. 3 marks, and I know this because I've learned my bacterium. I know how to label my diagrams. Okay, now the next question. How do you think this bacterium moves? Okay, so it's looking at the picture. What is going to give us an idea of how it moves? And it says here, in your answer, use evidence from the diagram. Okay, and it's telling you, you must use, is what in this diagram. So you're going to go here, and you're going to see that there is this structure over here. It's number E, and it is a flagellum. Now let's see how we answer it. Let's answer it in as it goes. How do you think this bacterium moves? You can tell me it swims. That is your first mark. And then it says, in your answer, use evidence from the diagram. Why? The bacterium has, right, I'm just going to write here. The bacterium has a flagella or a flagellum, right, however wave you've got there. So use, there we go, your second mark we're using from the diagram over there. Okay, this one now is going to be a calculation question. Now it says calculate the actual width of the cholera bacterium between points B and C. Give your answer in micrometers and show your working. Okay guys, so here we have the diagram of the bacterium, and we've been asked to find, right, the actual size between B, or let me get my pen going again. There we go, lovely blue, between B and C. Now I don't know if you guys know this, but I'm going to show you how to use the DAM triangle. All right, and I'm not, as I said, I'm not swearing. What does the DAM triangle mean? Okay, so when we look at the DAM triangle, the D stands for drawing. So if you measure a drawing or a picture that you have drawn, A is the actual size, and this is the one that we have to look. So this is a question mark. And M stands for magnification. So in order to do this, I need to have two bits of information. Okay, so let's have a look. So if I was to do the formula, I put the A equals. Now I'm going to quickly change my pen color because I want to show you just something that I'm going to use. This line over here means divide. Okay? This line between A and M means times. So if I go back to my formula, I want to find A because that's what it said, actual size. So I take D, which is my drawing, okay, and I divide it, because that's the divide line, over my magnification. Now I take my ruler, because I always take a ruler into the test, and I take my ruler and I measure this line over here from B to C, and I'm very accurate. And maybe it would be easier, because we have to convert it to microns to do it in millimeters. So say, for example, I measure from B to C, and I see that it is 50 millimeters. So I write down, okay, because that's what I measured. The magnification was given to me, okay. So there we go. I put there 50,000 times. And now I'm going to work out. So over here, 1, 2, all right, there's 1, so I only have to cross out 1, not the 2. So let's go back to that, take that out. And there's a reason why I don't teach maths. Okay, so 1 there, 1 there, all right, and then you take your calculator. You can use it anyway, or you can use it however you want. I don't even do it. Just take your calculator and you take 50 divided by 50,000. Don't try and do all of those funny little things. And you're going to get an answer, and because you measured in millimeters, you guys have got your calculator, you are going to give me your answer in millimeters. Okay, now you're not finished there. Now you need to convert it to microns. Now, that means you must take this answer and so you answer that you got, and you times it by 1,000. And that will be your final answer, and the units for microns or micrometers is that. Okay. Not so easy. I don't know if you've been taught the damn triangle. Maybe you know a different way in which you have been taught, but this is the way that I teach my kids, right? And sometimes it's easier if they just remember the damn triangle. Okay, guys, one more question. Okay, now here is a bit of an experiment that we've set up, and this is what it says. Okay, so now there's an agar plate, okay, and that is how we grow bacteria. And on that plate, with a culture of species of bacterium, usually found in the mouth. Okay, now we take 4 sterile. Sterile means that we've disinfected, they're pure, they've got nothing on them. And the paper discs A, B, C, and D, each containing a different brand of mouthwash. Now, you use mouthwash because it's a disinfectant. It kills bacteria in your mouth, okay? And they were then placed on the agar plate. The drawing shows the appearance of the plate after it had been incubated, right, at 37 degrees for 3 days. So have a look at this diagram, okay? All of these little dots is where they put bacteria on, okay? Then they took these plates of sterile plates. They were sterile. And what they did was they put the different mouthwashes on those little pieces of paper all around here. Okay, now remember, mouthwash is antibacterial. So what they wanted to see was they were looking for the biggest area where no bacteria was grown. So obviously, the biggest area where there was white, that there was no bacteria, meant that the mouthwash worked the best. Okay, so what question can we ask you over here? Right, first question. Explain why the paper discs, hang on a second, here we go. Which mouthwash is the most effective? Explain your reasoning. Okay, so which mouthwash had the most white area? So you're going to put there number C, so you get your 1 mark. And then it says, explain your reasoning. Why? And how did you explain it? You explained it. It had the largest area of no bacteria. Right, so that's what you're looking at. You grew this bacteria, and you now need to see, all right, why you did that. Okay, guys, one more question. I think then we're running out of time. Explain why the paper discs were sterile. So why did you have to put sterile paper discs there? Very important, all right? So no bacteria was growing on them. So they were pure, there was no growth, it wasn't going to interfere with your experiment at all. And if you notice, they grew it at 37 degrees, which was perfect temperature, body temperature. Okay, guys, that's all from us today. All right, hope you have a really good day, and we will see you next time.